In the quiet moments of rest, when the body settles into stillness and the mind loosens its grip on the day, an immense amount of activity continues beneath the surface. Life does not pause when awareness fades. Instead, it proceeds with a steady confidence, guided by systems so refined and reliable that they rarely call attention to themselves. This inner world is neither loud nor hurried. It is composed of countless small events unfolding in precise order, each one contributing to the simple fact of being alive. Within this hidden landscape, the body behaves less like a machine and more like a flowing process. Nothing operates in isolation. Every movement, every breath, every subtle adjustment depends on cooperation at levels far too small to be felt or seen. At the heart of this cooperation lies chemistry, not the sharp-edged chemistry of laboratories and equations, but a quiet biological chemistry that unfolds continuously from one moment to the next. The human body is made of matter, but it is sustained by change. Molecules are built, taken apart, reshaped, and exchanged in patterns that repeat with remarkable consistency. These changes do not happen by chance. They follow pathways that have been refined over long stretches of evolutionary time. The pathways themselves are calm, deliberate, and efficient, designed not for speed alone, but for balance and stability. In this gentle introduction, it helps to imagine the body not as a collection of organs or systems, but as a softly illuminated interior space, filled with motion that never becomes chaotic. Within this space, countless chemical reactions are occurring right now. Sugars are being transformed into energy. Proteins are being adjusted, repaired, or replaced. Signals are being passed from one cell to another, not with urgency, but with quiet precision. None of this would be possible if each chemical reaction were left to unfold on its own. Under ordinary conditions, many of the reactions essential to life would proceed far too slowly to support living tissues. Others might occur too quickly, releasing energy in ways that could damage delicate cellular structures. Life requires a middle path, one where reactions occur at just the right pace, neither rushed nor delayed. This is where enzymes enter the story, not as dramatic figures, but as patient facilitators. Enzymes are molecules, themselves products of the body's chemistry, that gently guide reactions along preferred routes. They do not force change. Instead, they lower barriers, smooth transitions, and allow chemical processes to unfold naturally within the narrow conditions compatible with life. It is important to understand that enzymes are not rare or exceptional. They are everywhere within the body. Each cell contains thousands of different enzymes, each with a specific role, each performing its task over and over again with quiet reliability. At this very moment, enzymes are helping your cells extract energy from nutrients, maintain the structure of your tissues, and preserve the internal balance that allows sleep to come at all. The remarkable thing about enzymes is not only what they do, but how effortlessly they do it. They are not consumed by the reactions they assist. After guiding one reaction, they remain ready to guide another, and another still. This capacity for repeated service is one of the reasons life can persist without constant replenishment of its molecular tools. From a broader perspective, enzymes allow the body to exist in a world that would otherwise be chemically inhospitable. The temperatures at which humans live are mild, far below those that would naturally drive many chemical reactions forward. Without enzymes, the chemistry of life would either stall or require conditions incompatible with delicate biological structures. Enzymes solve this problem quietly, enabling life to flourish within narrow and comfortable limits. As you rest, your body temperature remains carefully regulated. Your blood chemistry stays within narrow ranges. Your cells continue to repair themselves, replacing worn components with new ones. None of these processes require instruction from the conscious mind. They unfold automatically, guided by networks of enzymes responding to local conditions within tissues and cells. In this sense, enzymes represent a form of biological wisdom. They respond not to thoughts or intentions, but to the immediate chemical environment. When a particular molecule becomes abundant, enzymes that act upon it become more active. When levels fall, activity slows. This responsiveness allows the body to adjust continuously, moment by moment, without overshooting or delay. The calm reliability of enzymes is especially evident during sleep. As external demands fade, internal maintenance becomes more prominent. Cells use this time to restore balance, clear waste products, and reinforce the structures that will be needed when wakefulness returns. Enzymes oversee these restorative processes with the same quiet dedication they bring to every other moment. It can be comforting to know that life is supported not by constant vigilance, but by deeply ingrained chemical patterns that require no supervision. Even as consciousness drifts, the inner world remains attentive in its own way. The reactions that sustain life continue to flow, guided by molecular forms shaped over millions of years. Throughout this series, the focus will remain on these silent contributors to existence. There will be no need for urgency or complexity beyond what is necessary to understand their role. Biochemistry, at its core, is not about speed or pressure. It is about order, balance, and continuity. Enzymes embody these qualities in their purest form. For now, it is enough to hold a simple image. Within you, an unseen community of molecules is working steadily, ensuring that life proceeds smoothly from one moment to the next. There is no need to follow each reaction or remember each name. The purpose here is not mastery, but appreciation. As the body rests, enzymes continue their quiet labor, guiding the chemistry that keeps cells alive, tissues functional, and systems in harmony. This calm, persistent activity forms the foundation upon which everything else is built. 2. The nature of enzymes. To understand enzymes, it helps to begin from a place of simplicity. At their core, enzymes are molecules, no different in substance from many others found within the body. They are not alive in themselves, nor do they possess intention or awareness. And yet, without them, life as we experience it could not continue. Their importance lies not in complexity for its own sake. Enzymes belong to a broad class of biological molecules known as proteins. Proteins are assembled from smaller units called amino acids, linked together in long chains that fold into precise shapes. It is this folding, this quiet arrangement of form, that gives enzymes their remarkable abilities. Each enzyme's shape allows it to interact with other molecules in a very specific way, much like a familiar gesture recognized without thought. In the body, countless chemical reactions are constantly possible. Molecules collide, drift apart, and rearrange themselves according to the laws of chemistry. Left on their own, however, many of the reactions essential to life would occur either far too slowly or not at all under the gentle conditions found inside living cells. Enzymes provide a solution that is both elegant and efficient. They create a pathway that allows reactions to proceed smoothly, without the need for extreme heat, pressure, or energy input. This guiding role is why enzymes are often described as catalysts. A catalyst is something that speeds up a chemical reaction without being consumed by it. In this sense, enzymes are not participants in the reaction's outcome, but facilitators of its journey. After a reaction is complete, the enzyme remains unchanged, ready to assist again. This ability to function repeatedly contributes to the quiet endurance of biological systems. What makes enzymes especially suited to life is their selectivity. Each enzyme typically assists only a narrow range of reactions, often just one specific transformation. This specificity prevents chemical chaos. It ensures that molecules are altered in controlled ways, at controlled times, and in controlled places. Within the dense environment of the cell, where thousands of different molecules are present at once. Such precision is essential. The specificity of enzymes arises from their structure. The folded shape of an enzyme creates a region where other molecules can briefly attach. This region, often described as an active site, is shaped in such a way that only certain molecules can interact with it comfortably. When the appropriate molecule encounters the enzyme, the two fit together momentarily, forming a temporary partnership that allows the reaction to proceed. This interaction is not rigid or forceful. It is dynamic and flexible, allowing small adjustments as the reaction unfolds. Once the transformation is complete, the product molecules drift away, leaving the enzyme free to resume its role. Throughout this process, the enzyme remains intact, unchanged by the work it has performed. It is important to note that enzymes do not create reactions out of nothing. They do not override the fundamental laws of chemistry. Instead, they lower the energetic barriers that normally slow reactions down. By stabilizing transitional states and guiding molecules along favorable paths, enzymes make it easier for reactions to occur under the mild conditions of the living body. This gentle assistance allows life to exist within a narrow range of temperature and chemical balance. Human enzymes, for example, are finely tuned to operate at around normal body temperature. Outside this range, their structure can become unstable. This sensitivity underscores how closely enzyme function is tied to the broader stability of the internal environment. Despite this sensitivity, enzymes are remarkably reliable. There is no central command directing each enzyme when to act. Instead, activity emerges naturally from local conditions. When the molecules involved in a reaction are present, enzymes respond. When those molecules are scarce, activity slows. This responsiveness creates a form of chemical self-regulation. The body does not need to anticipate every change or prepare explicit instructions. Enzymes respond in real time to what is available, adjusting the pace of reactions moment by moment. This is one of the reasons living systems are so resilient. They are flexible without being fragile. Another defining feature of enzymes is their efficiency. A single enzyme molecule can assist thousands, sometimes millions, of reactions per second. This rapid turnover allows cells to sustain high levels of activity without needing enormous quantities of catalytic material. The economy of this system is one of its quiet strengths. Enzymes are also deeply interconnected. Rarely does one enzyme act in isolation. Instead, they function in sequences, where the product of one reaction becomes the starting point for the next. These sequences form pathways, and pathways combine into networks that span entire cells and tissues. Within these networks, enzymes pass molecular materials along like gentle couriers, each one contributing a small but essential step. Because of this interconnectedness, changes in enzyme activity can ripple outward. A slowdown in one reaction can influence many others downstream. The body manages this complexity through layers of regulation, ensuring that fluctuations remain within safe limits. Once again, enzymes are central to this regulation, both as the agents of change and as the points of control. From an evolutionary perspective, enzymes represent solutions refined over immense spans of time. Early life depended on simpler chemical processes, but as biological systems grew more complex, the need for precise control increased. Enzymes provided a way to manage this complexity without sacrificing stability. Those enzyme structures that worked reliably were preserved and refined, becoming the foundations of modern biochemistry. In humans, many enzymes are shared with other forms of life. The enzymes that help manage energy, repair DNA, or build proteins are often strikingly similar across species. This shared chemistry speaks to a deep unity underlying life on Earth. It also reflects the effectiveness of enzymatic solutions once they emerge. As you rest and breathe, enzymes continue their work without pause. They do not tire or lose focus. Their activity is woven into the fabric of existence itself. Understanding their nature is not about memorizing names or pathways, but about appreciating the quiet logic that allows life to persist. In the next part, attention will turn toward the broader chemical flow that enzymes support, exploring how life itself can be understood as a continuous, ordered movement of matter and energy. For now, it is enough to recognize enzymes as steady guides, shaping the chemistry that keeps the inner world calm, balanced, and alive. 3. Life is a flow of chemistry. Life, when viewed from a quiet distance, is less a collection of solid parts and more a continuous movement. Even the most stable structures in the body are in subtle transition, shaped and reshaped by chemistry that never fully comes to rest. What we call being alive is not a fixed state, but an ongoing process, a gentle flow of matter and energy guided along predictable paths. At every moment, molecules within the body are interacting. Some join together, others separate, and still others change form entirely. These transformations follow physical laws that apply everywhere in the universe, yet within living systems they are arranged into patterns that sustain structure, function, and continuity. This organization is what distinguishes living chemistry from the chemistry of the non-living world. In a single cell, thousands of chemical reactions are taking place at once. Sugars are being converted into usable energy. Amino acids are being assembled into proteins. Lipids are being adjusted to maintain membranes. None of these reactions occur in isolation. Each is connected to others, forming chains and loops that allow the cell to adapt to changing conditions while maintaining internal balance. This interconnectedness creates a sense of flow. Materials enter a cell, are transformed through a series of steps. Energy moves through these pathways as well, captured, stored briefly, and released in controlled amounts. The result is not turbulence, but steadiness, a chemical current that sustains life without drawing attention to itself. One way to understand this flow is to think of metabolism, the sum of all chemical reactions occurring within the body. Metabolism is not a single process, but a vast network of pathways, each composed of many individual steps. Some pathways build molecules up, others break them down, and many do both, depending on the body's needs at a given moment. These pathways are carefully balanced. When nutrients are abundant, chemical reactions favor storage and repair. When nutrients are scarce, reactions shift toward release and conservation. This flexibility allows the body to respond to daily rhythms, seasonal changes, and periods of rest or activity without losing stability. The flow of chemistry is not random, it is directional, shaped by gradients of energy and concentration. Molecules tend to move from areas of higher concentration to lower concentration, from higher energy states to lower ones. Enzymes help guide this movement, ensuring that energy is released gradually rather than all at once, and that molecular changes occur in useful directions. Within this flowing system, time takes on a special role. Reactions must occur in the correct sequence. If one step happens too soon or too late, the entire pathway can be disrupted. Enzymes help maintain proper timing, coordinating reactions so that products appear when and where they are needed. This coordination allows the body to operate efficiently. Energy is not wasted, and harmful byproducts are minimized. Chemical intermediates that might be unstable or reactive are quickly passed along to the next step in a pathway, reducing the chance of unintended reactions. In this way, flow becomes protection as well as function. The idea of life as chemical flow also helps explain why living systems require constant input. Food, oxygen, and water provide the raw materials and energy needed to sustain ongoing reactions. Without this input, the flow gradually slows, pathways falter, and the organization that defines life begins to fade. Yet even in the absence of conscious activity, this flow persists. During sleep, When muscles relax and external engagement diminishes, internal chemistry continues with quiet determination. Energy is redirected toward repair and maintenance. Waste products are processed and removed. Chemical gradients are restored in preparation for waking. This nighttime chemistry is not separate from daytime function. It is part of the same continuous process, unfolding across hours, days, and years. The boundaries we draw between activity and rest are meaningful to experience, but chemically, the body remains in motion throughout. At the smallest scale, individual molecules participate in this flow without awareness or intent. They follow the contours of energy landscapes shaped by enzyme structures and cellular environments. At a larger scale, the combined effect of countless molecular interactions gives rise to the stable patterns we recognize as health and vitality. One of the remarkable features of this system is its tolerance for variation. The exact number of molecules involved in a reaction may fluctuate. Reaction rates may speed up or slow down slightly, yet the overall flow remains steady. This robustness arises from redundancy and regulation built into metabolic networks. If one pathway slows, another may compensate. If demand increases in one area, resources are redirected from elsewhere. Enzymes respond dynamically to these shifts, altering their activity in response to chemical signals. This adaptability allows life to persist in environments that are never perfectly constant. The flow of chemistry also connects the inner world of the body to the outer world. Molecules that enter through food or air are gradually incorporated into tissues, circulated, transformed, and eventually return to the environment. Atoms that once belonged to plants, animals, or the atmosphere become part of you for a time, participating in your unique chemical patterns before moving on. This perspective can be calming. It reminds us that the body is not an isolated entity, but part of a larger exchange. The chemistry that sustains life is shared across ecosystems and generations. The same fundamental reactions that occur within you have been occurring in one form or another since life began. Understanding life as a flow rather than a static structure also reframes change. Growth, aging, and adaptation are not abrupt events, but gradual shifts in chemical balance and pathway activity. Enzymes adjust their roles as conditions change, supporting new patterns while letting old ones fade. Throughout this continuous motion, balance remains the guiding principle. Not a rigid balance that resists all change, but a dynamic equilibrium that allows movement without collapse. Enzymes help maintain this equilibrium by ensuring that reactions proceed smoothly and reversibly where needed. As you rest now, this chemical flow continues beneath awareness. There is no need to monitor it or intervene. The pathways that sustain you have been refined to operate reliably on their own. Life moves forward, moment by moment, guided by chemistry that is both precise and gentle. In the next part, attention will turn more closely to why these reactions require guidance at all, and how enzymes make it possible for chemistry to unfold at the calm pace required for living systems. For now, it is enough to recognize that being alive means being in motion, carried along by a quiet, continuous flow that does not pause, even in sleep. 4. Why reactions need guidance. Within the body, chemistry unfolds under conditions that are gentle by any physical standard. Temperatures remain moderate, pressures are low, and the environment is mostly water. These are comfortable conditions for living tissues, but they are not conditions under which most chemical reactions naturally proceed with ease. In laboratories, chemists often rely on heat, strong acids, or high pressures to drive reactions forward. The body cannot afford such extremes. Its chemistry must operate quietly, safely, and continuously, without damaging the structures it depends on. At the heart of this challenge lies a simple reality. Many chemical reactions require an initial push. Molecules are stable arrangements of atoms, and changing those arrangements often involves passing through a brief, unstable state. This transitional moment requires energy. Without sufficient energy, reactions proceed very slowly, sometimes so slowly that for biological purposes, they might as well not occur at all. In the absence of guidance, molecules rely on random collisions. They drift through the watery interior of the cell, occasionally bumping into one another. Most of these encounters are unproductive. Either the molecules are not oriented correctly, or they lack the energy needed to rearrange their bonds. Left to chance alone, the chemistry of life would be sluggish and unreliable. This is where guidance becomes essential. The body needs a way to help molecules overcome these energetic barriers without resorting to harsh conditions. It needs reactions to occur quickly enough to support life, but gently enough to preserve delicate cellular structures. Enzymes provide this guidance in a manner that is both precise and restrained. Rather than forcing reactions to happen, enzymes reshape the path that reactions take. This is often described as lowering the activation energy of a reaction. In practical terms, it means that molecules can change form more easily, even at normal body temperature. This guidance is subtle. Enzymes do not supply energy themselves. Instead, they stabilize transitional states, holding molecules in positions that make rearrangement easier. By reducing the energetic cost of reaching these states, enzymes allow reactions to proceed smoothly and predictably. The need for such guidance becomes clearer when considering the sheer number of reactions taking place simultaneously. In a single cell, thousands of reactions may be occurring at any given moment. Each must happen in a specific order and at a specific rate. Without control, reactions could interfere with one another, producing unwanted byproducts or consuming resources inefficiently. Guidance also prevents dangerous shortcuts. Some chemical reactions, if allowed to proceed unchecked, could release energy too quickly or produce reactive intermediates that damage cellular components. Enzymes help channel reactions along safer pathways, ensuring that energy is released gradually and products are stable. This controlled release of energy is especially important for processes like metabolism. When the body extracts energy from nutrients, it does so step by step. Each step releases only a small amount of energy, which can be captured and used effectively. Without enzymatic guidance, energy might be released all at once, generating heat rather than useful work. The need for guidance is not limited to energy metabolism. Building molecules also requires careful control. Proteins, nucleic acids, and other complex structures are assembled through sequences of reactions that must occur in the correct order. Enzymes ensure that each building step happens at the right time and place, reducing errors and waste. Another reason reactions need guidance is specificity. The interior of the cell is crowded with molecules of many different types. If reactions occurred indiscriminately, molecules might interact in unintended ways. Enzymes provide selectivity, bringing together only the molecules involved in a particular reaction. This reduces confusion and preserves the integrity of cellular processes. This specificity also allows the same basic chemical building blocks to be used in multiple contexts. The same types of atoms and bonds appear throughout the body, yet they are arranged into a wide variety of molecules. Enzymes direct these arrangements, determining which bonds form and which are broken. Shaping diversity from simplicity, guidance further allows the body to regulate its chemistry in response to changing conditions. When demand for a particular product increases, enzymes involved in its production can become more active. When demand decreases, activity slows. This responsiveness ensures that resources are allocated efficiently, without excess buildup or depletion. The guidance provided by enzymes is local rather than centralized. There is no single control center overseeing every reaction. Instead, Control emerges from the properties of enzymes themselves and the conditions within the cell. This decentralized approach adds resilience. If one pathway is disrupted, others can adjust to compensate. It is also worth noting that not all reactions require the same degree of guidance. Some chemical changes occur readily under biological conditions, while others are inherently difficult. Enzymes are especially important for these challenging reactions, making possible transformations that would otherwise be impractical within living systems. The quiet effectiveness of enzymatic guidance often goes unnoticed because it is so reliable. We rarely experience the absence of this guidance directly. When it does fail, however, the consequences can be significant. Disruptions in enzyme function can slow critical reactions or allow harmful ones to proceed, highlighting how dependent life is on these molecular guides. Yet under normal conditions, enzymes perform their roles without drama. They work continuously, adjusting to subtle changes, maintaining the flow of chemistry with minimal fluctuation. This steadiness contributes to the sense of continuity we associate with being alive. As you rest, this guided chemistry continues. Reactions proceed at the pace they need, neither rushed nor delayed. Energy is released gently, structures are maintained, and balance is preserved. There is no need for conscious oversight. In the next part, attention will shift toward the relationship between molecular shape and function, exploring how the physical form of enzymes enables them to guide reactions so precisely. For now, it is enough to recognize that life depends not just on chemistry, but on chemistry carefully guided, softened, and shaped to fit the quiet conditions of the living body. 5. Shape, fit, and molecular calm. At the smallest scales of life, shape is not an aesthetic detail but a quiet form of instruction. The way a molecule folds, bends, and presents its surface determines how it will behave, what it can interact with, and what role it will play within the larger flow of chemistry. Enzymes are perhaps the clearest expression of this principle. Their function arises directly from their form, and their form is maintained with remarkable consistency under the calm conditions of the living body. An enzyme begins its existence as a chain of amino acids, assembled according to instructions encoded in DNA. This chain is not meant to remain extended or linear. As soon as it is formed, it begins to fold inward on itself, guided by subtle chemical attractions between its components. Portions of the chain are drawn together, others are repelled, and gradually a stable three-dimensional structure emerges. This folded shape is not arbitrary. It is the result of countless small interactions, settling into a configuration that is energetically favorable and functionally precise. Within this folded structure, certain regions become especially important. These areas form pockets, grooves, or surfaces that can interact with other molecules. The overall enzyme may be large compared to the molecules it works on, but only a small portion is directly involved in guiding the reaction. This region provides a space where another molecule can settle briefly, held in place by a gentle network of chemical forces. The idea of fit is often used to describe this interaction, but it is important to imagine fit not as something rigid, but as something responsive. Neither the enzyme nor the molecule it binds is perfectly fixed. Both retain a degree of flexibility, allowing them to adjust slightly as they come together. This flexibility contributes to calm rather than strain, allowing interactions to occur without force. When a molecule enters the enzyme's active region, it is not locked into place. Instead, it is guided into an orientation that makes the next chemical step easier. Bonds may be stretched slightly, angles adjusted, or charges redistributed. These changes lower the energy required for the reaction to proceed, smoothing the path from one form to another. This shaping effect is subtle but powerful. By holding molecules in favorable positions, enzymes reduce the randomness of chemical collisions. They replace chance with structure, without eliminating freedom entirely. Molecules still move, still fluctuate, but within a framework that makes productive interactions more likely. The calm efficiency of this process is striking. Enzymes do not rush reactions forward. They simply make the desired transformation the easiest option available. In doing so, they allow chemistry to proceed at a pace compatible with life, neither stalled nor uncontrolled. The relationship between shape and function also explains why enzymes are sensitive to their environment. Temperature, acidity, and chemical composition all influence the forces that hold an enzyme's shape together. Under stable conditions, these forces maintain the enzyme's structure with ease. When conditions drift too far from the optimal range, the balance can be disturbed, and the enzyme may lose its precise form. This sensitivity is not a weakness but a reflection of fine-tuning. Enzymes are designed to operate within the narrow conditions maintained by living systems. The same regulatory mechanisms that keep body temperature and internal chemistry stable also preserve enzyme structure. In this way, the body protects its molecular guides by maintaining a calm internal environment. The concept of molecular calm extends beyond individual enzymes. Within cells, enzymes are often arranged in sequences or complexes that further enhance efficiency. When one enzyme completes its task, the product may pass directly to the next enzyme in line. This reduces unnecessary movement and keeps reactive molecules from drifting away or causing unintended reactions. Such arrangements create microenvironments where chemistry unfolds with minimal disruption. The cell becomes a space where molecules move purposefully, guided not by conscious intent, but by physical form and chemical compatibility. The overall effect is one of order emerging naturally from structure. Shape also allows enzymes to discriminate between very similar molecules. Two molecules may differ by only a small feature, yet an enzyme can distinguish between them reliably. This discrimination ensures that the correct reactions occur, even in a crowded cellular environment. Without such selectivity, the body's chemistry would lose its coherence. This precision supports the broader stability of metabolic pathways. Each step produces exactly the molecule needed for the next, reducing waste and preventing accumulation of unwanted intermediates. The flow of chemistry remains smooth. With minimal turbulence, it is worth reflecting on how this molecular precision contributes to the feeling of continuity we associate with life. Despite constant molecular turnover, tissues maintain their identity. Cells replace components without losing function. This stability arises because enzymes shape change in consistent ways, preserving overall patterns even as individual molecules come and go. As you rest, these processes continue quietly. Enzymes maintain their shapes, molecules find their places, and reactions proceed with gentle regularity. There is no sensation associated with this activity, no signal sent to consciousness. Yet it is this unseen choreography that allows rest to be restorative and sleep to be safe. The calm nature of enzyme function also reflects a deeper principle of biological design. Life does not rely on extremes. It favors moderation, balance, and efficiency. By shaping reactions rather than forcing them, enzymes exemplify this approach. They guide rather than command. From a broader perspective, the relationship between shape and function extends to all levels of biology. Cells, tissues, and organs all depend on form to guide activity. At the molecular level, enzymes are simply the most refined expression of this principle, operating where precision matters most. As this exploration continues, future parts will look at how these shaped interactions contribute to stability across entire systems, and how enzymes participate in maintaining balance in changing conditions. For now, it is enough to recognize that, within you, countless molecular forms are holding steady, allowing chemistry to proceed calmly, reliably, and without interruption. The body rests, the mind drifts, and beneath it all, shape guides function, ensuring that life continues with quiet assurance through every moment of sleep. 6. Enzymes and stability. Stability within the living body is not a matter of stillness, but of balance maintained through constant adjustment. At every moment, conditions shift slightly. Molecule concentrations rise and fall. Temperatures fluctuate by small degrees, and energy demands change as activity and rest alternate. Yet, despite these variations, the internal environment remains remarkably steady. This steadiness is known as homeostasis, and enzymes play a central role in preserving it. To appreciate this role, it helps to think of stability not as a fixed point, but as a range within which life can comfortably exist. Blood chemistry, cellular conditions, and energy availability all vary within narrow limits. When values drift toward the edges of these limits, corrective processes quietly come into play. Enzymes are woven deeply into these corrective responses, acting as both sensors and agents of change. Because enzyme activity depends on the surrounding chemical environment, enzymes naturally respond to shifts in that environment. When the concentration of a particular molecule increases, enzymes that act upon it encounter it more frequently. As a result, the rate of reaction increases without any external instruction. When concentration decreases, reaction rates slow just as naturally. This simple relationship allows enzyme-driven processes to self-regulate with remarkable precision. This responsiveness creates a buffering effect. Sudden increases in certain substances are met with increased processing. Sudden decreases lead to conservation. In this way, enzymes help smooth out fluctuations, preventing sharp swings that could disrupt cellular function. The chemistry of life remains calm, even when inputs change. Stability also depends on timing. Some reactions must occur rapidly in response to immediate needs, while others unfold slowly over hours or days. Enzymes support this temporal balance by differing in how readily they become active. Some are always present and active, maintaining essential background processes. Others remain relatively inactive until conditions change, at which point their activity increases. This layered arrangement allows the body to prioritize. Essential processes continue without interruption, while flexible processes adjust according to circumstance. Enzymes enable this prioritization through their sensitivity to local signals, such as changes in molecule concentration, acidity, or energy availability. Another aspect of stability is prevention. Many potentially harmful chemical reactions are possible within the cellular environment. Reactive molecules can damage proteins, lipids, or genetic material if left unchecked. Enzymes help prevent this damage by rapidly converting reactive intermediates into more stable forms, or by directing them into controlled pathways where their energy can be safely managed. In this way, enzymes act as quiet guardians of cellular integrity. They do not eliminate risk entirely, but they reduce it to levels compatible with long-term survival. The cumulative effect of these protective actions contributes to the resilience of living systems. Stability also relies on coordination across different pathways. Energy production, nutrient processing, waste removal, and repair mechanisms must remain in harmony. If one pathway accelerates while another lags, imbalance can result. Enzymes help synchronize these pathways by responding to shared signals and shared resources. For example, when energy demand increases, enzymes involved in energy-releasing pathways become more active. At the same time, enzymes involved in energy storage may reduce their activity. This coordinated response ensures that energy is made available when needed, without unnecessary accumulation or depletion. The elegance of this system lies in its simplicity. Rather than relying on centralized control, stability emerges from local interactions. Each enzyme responds to its immediate environment, and the combined effect of many such responses produces a stable whole. This decentralized approach allows for flexibility and redundancy, making the system robust against minor disruptions. As you rest, stability becomes especially important. During sleep, the body shifts its priorities. Energy is redirected from movement and perception toward repair and maintenance. Enzymes adjust their activity accordingly, slowing some pathways and accelerating others. This shift occurs gradually and smoothly, without abrupt transitions. Throughout the night, enzymes support processes such as protein repair, membrane maintenance, and the clearing of metabolic byproducts. These activities restore balance at the cellular level, preparing the body for the next period of wakefulness. The calm rhythm of enzyme activity mirrors the rhythm of sleep itself. It is worth noting that stability does not mean uniformity. Different tissues maintain slightly different conditions suited to their functions. Enzymes within each tissue are adapted to these local environments, ensuring that stability is achieved in context-specific ways. What remains consistent is the principle of balance through guided chemistry. Over longer timescales, enzymes also contribute to stability across the lifespan. As the body grows, matures, and ages, enzyme activity patterns shift gradually. Some pathways become less active, others more so. These changes reflect adaptation rather than failure, allowing the body to function effectively under changing conditions. This adaptability underscores another dimension of stability: persistence through change. Enzymes enable living systems to maintain identity and function, even as individual components are replaced. Proteins turn over, cells renew themselves, yet the overall pattern remains recognizable. Stability, in this sense, is continuity rather than permanence. As consciousness fades and sleep deepens, there is comfort in knowing that stability does not depend on vigilance. Enzymes continue their quiet work, responding to shifts, correcting deviations, and maintaining the internal environment with gentle precision. In the next part, attention will move toward the role of enzymes in digestion, where stability must be maintained even as the body processes materials from the outside world. For now, it is enough to rest in the understanding that beneath the surface of sleep, a calm network of molecular guides is preserving balance, ensuring that life continues steadily, moment by moment, through the night. 7 digestive enzymes at work. Every day, the body encounters the outside world in one of the most intimate ways possible. What begins as something solid, complex, and unfamiliar must be transformed into simple, usable forms that can enter the body's internal chemistry. This transformation is not abrupt or forceful. It is gradual, methodical, and calm, guided almost entirely by enzymes that specialize in digestion. Digestion is often imagined as a process of breaking things apart, but in reality it is a careful sequence of changes. Large, complex molecules are guided, step by step, into smaller units, each step controlled to prevent damage and waste. Digestive enzymes ensure that this transformation happens efficiently, safely, and in harmony with the body's overall stability. The process begins even before food reaches the stomach. As food enters the mouth, enzymes already present in saliva begin their work. These enzymes do not attempt to complete digestion. Instead, they initiate it, gently preparing certain components for the longer journey ahead. This early involvement reflects a broader pattern in digestion. Each stage contributes a part, never rushing, never overwhelming the system. As food moves through the digestive tract, it encounters different environments, each with its own conditions. Changes in acidity, moisture, and movement help enzymes do their work more effectively. Digestive enzymes are adapted to these environments, functioning optimally where they are needed and becoming inactive where they are not. This ensures that reactions occur in the correct place and sequence. In the stomach, digestion becomes more intensive, but still controlled. Enzymes here are specialized to work in acidic conditions, guiding the transformation of proteins into smaller fragments. The acidity itself helps unfold complex molecules, making them more accessible to enzymatic action. Yet even in this active environment, digestion proceeds steadily rather than violently. As partially digested material moves onward, it enters a region where digestion reaches its most refined stage. Here, a wide array of enzymes collaborate to complete the breakdown process. Each enzyme focuses on a specific type of molecule, ensuring that carbohydrates, proteins, and fats are all handled appropriately. This division of labor prevents confusion and maintains efficiency. What is remarkable about this system is its adaptability. When certain nutrients are more abundant, the enzymes that process them become more active. When those nutrients are scarce, activity diminishes. This responsiveness helps maintain balance, preventing excess accumulation of any one component. Throughout digestion, enzymes act without drawing attention to themselves. There is no sensation associated with their work under normal conditions. The process is quiet, unfolding beneath awareness, integrated seamlessly into the body's daily rhythm. This calm efficiency allows digestion to coexist comfortably with other processes, such as rest and sleep. Once digestion has reduced food into its basic building blocks, enzymes continue to play a role in preparing these molecules for absorption. Sugars, amino acids, and fatty components must be converted into forms that can cross cellular barriers safely. Enzymes guide these final adjustments, ensuring that nutrients enter circulation smoothly. The digestive system is also careful not to digest itself. Enzymes are produced in inactive forms or confined to specific regions until they are needed. This precaution reflects the importance of control. Digestive enzymes are powerful tools, and their activity must be precisely regulated to protect the tissues that produce them. This regulation is achieved through a combination of physical separation, chemical conditions, and timing. Enzymes become active only when they reach the correct environment. Once their task is complete, they are broken down or removed, preventing unnecessary activity. The result is a system that is both effective and safe. Digestive enzymes also contribute to stability beyond nutrient processing. By breaking down food into predictable components, they ensure that the body receives a consistent supply of raw materials. This consistency allows other metabolic pathways to function reliably, supporting energy production, repair, and growth. During sleep, digestion does not stop, but it slows. Nutrients absorbed earlier are processed and distributed, and digestive tissues themselves undergo maintenance and repair. Enzymes guide these nighttime processes with the same calm precision they bring to active digestion. It is worth noting that digestive enzymes reflect a long evolutionary history. The ability to extract nourishment from diverse food sources has been essential for survival. Over time, enzymes that could handle a wide range of molecules were refined and preserved. This versatility allows humans to thrive on varied diets while maintaining internal balance. The calm, stepwise nature of digestion also illustrates a broader theme in biology: complexity managed through simplicity. Rather than relying on a single, overwhelming reaction, digestion is divided into many small, manageable steps. Each enzyme contributes a little, and together they achieve what no single reaction could accomplish safely. As you rest, the digestive enzymes that have been active throughout the day continue their quiet work. Some are completing reactions, others are being recycled, and still others are being prepared for future use. There is no urgency, only continuity. In the next part, attention will turn toward what happens after digestion is complete, exploring how enzymes help transform absorbed nutrients into forms that can be used by cells throughout the body. For now, it is enough to recognize digestion as a calm collaboration between structure, environment, and enzymatic guidance, one that supports life quietly, reliably, and without demand for attention, even as sleep deepens and awareness fades. 8. Absorption and preparation. Once digestion has gently reduced food into its simplest components, a quieter but equally important phase begins. The body must now decide what to keep how to use it, and where to send it. This stage is less about breaking down and more about refinement and preparation, ensuring that the products of digestion are smoothly integrated into the body's internal chemistry. Enzymes remain central to this process, guiding molecules as they move from the digestive tract into the broader circulation of life. Absorption is not a sudden transfer. It is a gradual crossing, where nutrients pass from the interior of the digestive system into the bloodstream and lymphatic pathways. The surfaces involved in this exchange are finely structured, increasing contact and slowing movement so that enzymes can complete their work with care. The goal is not speed, but accuracy and balance. Before many nutrients can be absorbed, they must be adjusted one final time. Sugars may be rearranged into specific forms, amino acids checked and modified, and lipid components repackaged. Enzymes oversee these adjustments, ensuring that molecules are compatible with the transport systems that will carry them onward. This preparation reflects a broader principle in biology. Materials are rarely used in the exact form in which they arrive. Instead, they are reshaped to fit the needs of the internal environment. Enzymes provide the means for this reshaping, allowing the body to accept a wide variety of inputs while maintaining internal consistency. Once nutrients cross into circulation, enzymes continue to guide their fate. Others are directed toward storage, to be held in reserve for later use. Still, others become building blocks for repair and renewal. These decisions are not conscious, but chemical, shaped by enzyme activity responding to signals of demand and availability. The liver plays a particularly important role in this stage of preparation. It acts as a chemical processing center, receiving nutrients soon after absorption and adjusting them as needed. Enzymes within liver cells transform sugars into storage forms, convert amino acids into useful intermediates, and modify fats for safe transport. Through these actions, the liver helps smooth out fluctuations in nutrient supply. This smoothing effect contributes to overall stability. Rather than allowing nutrient levels to spike and fall sharply after meals, enzymatic processing spreads availability over time. The body experiences nourishment as a steady presence rather than a series of abrupt changes. This steadiness supports calm function across all systems. Absorption and preparation also involve careful selectivity. Not everything that enters the digestive tract is allowed into the internal environment. Enzymes participate in processes that neutralize or modify potentially harmful substances, reducing their impact before they can circulate widely. This protective role adds another layer of stability to the system. As nutrients are prepared for use, enzymes ensure compatibility between pathways. A molecule destined for energy production must be shaped differently than one destined for structural repair. Enzymes handle these distinctions quietly, preventing confusion and misdirection. The result is an orderly distribution of resources that supports the body's immediate and long-term needs. During periods of rest, including sleep, this preparatory work continues. Nutrients absorbed earlier in the day are still being processed, sorted, and allocated. Enzyme activity shifts toward maintenance and storage, reinforcing reserves and supporting slow repair processes. There is no rush, only continuation. It is during these quieter hours that the value of enzymatic preparation becomes especially clear. By managing resources calmly and efficiently, enzymes allow the body to recover without strain. Cells receive what they need when they need it, without drawing on conscious effort or attention. Absorption and preparation also reflect a deep trust in timing. The body does not attempt to use everything at once. Instead, it relies on enzymatic pathways to release resources gradually, matching availability with demand. This trust allows systems to remain balanced even when external conditions change. From an evolutionary perspective, this stage of metabolism has been essential for survival. Organisms that could store and redistribute nutrients effectively were better equipped to endure periods of scarcity. Enzymes provided the means for this adaptability, allowing life to persist through changing environments. As you rest, the products of past meals are still finding their places. Some are reinforcing tissues, others are replenishing energy stores, and others are being quietly recycled. Enzymes oversee this distribution with steady patience, ensuring that nothing is wasted and nothing is overwhelmed. In the next part, attention will turn toward how prepared nutrients are transformed into usable energy at the cellular level, exploring the enzymatic processes that release energy in controlled, life-sustaining ways. For now, it is enough to recognize absorption and preparation as a calm bridge between the outside world and the inner chemistry that keeps you alive, working continuously and quietly as sleep deepens and the body rests in balance. 9 enzymes that release energy. The body, though seemingly still at rest, is engaged in a constant, subtle dance of energy. Cells are never fully at rest. Even when the mind is asleep, they are constantly working, repairing, and maintaining, using energy to fuel these processes, even during the quietest moments. The flow of energy throughout the body is orchestrated by enzymes, which guide the transformation of food into usable energy. This energy, once unlocked, powers the delicate machinery of life, allowing everything from cell repair to the continuous functioning of the heart and lungs. When you consume food, it is not immediately converted into usable energy. These enzymes help break down complex carbohydrates, fats, and proteins into smaller components, and eventually, these components are transformed into the energy molecules that sustain life. The energy molecule most directly involved in this process is ATP, or adenosine triphosphate. ATP is often described as the currency of cellular energy. It provides the power needed for nearly every cellular process, from muscle contraction to protein synthesis. The process begins in the digestive system, where food is broken down into its basic building blocks. Carbohydrates are reduced to sugars, proteins to amino acids, and fats to fatty acids and glycerol. These components are then absorbed into the bloodstream, and they make their way to cells where they can be further processed. However, before they can be used to generate energy, they must be transformed into simpler molecules that can enter the energy-producing pathways within the cell. The first major step in this process occurs in the cytoplasm of the cell. Here, glucose, one of the most common energy sources, is broken down through a process called glycolysis. Glycolysis involves a series of enzymatic reactions that split glucose into 2 molecules of pyruvate. This process, while it does not require oxygen, produces a small amount of ATP, which can be used immediately by the cell. Glycolysis is just the beginning. The pyruvate produced during this initial breakdown is transported into the mitochondria, the energy powerhouses of the cell. Here, pyruvate undergoes further transformation, entering the citric acid cycle, also known as the Krebs cycle. The citric acid cycle is a series of enzymatic reactions that takes place in the mitochondria, where the pyruvate is fully oxidized to release carbon dioxide and energy in the form of high-energy electrons. These electrons are then passed through the electron transport chain, another enzymatic pathway, where their energy is used to pump protons across the mitochondrial membrane, creating a proton gradient. It is this proton gradient that drives the final step in energy production, the synthesis of ATP. The enzyme ATP synthase uses the energy stored in the proton gradient to produce ATP from ADP, adenosine diphosphate, and inorganic phosphate. This process, called oxidative phosphorylation, is the most efficient way the body generates ATP, providing the energy that fuels most cellular activities. While this process primarily involves glucose, it is not limited to carbohydrates. Fats and proteins can also be used as energy sources, though their pathways are slightly different. Fatty acids, for example, are broken down into smaller units called acetyl-CoA molecules, which enter the citric acid cycle directly. Proteins, on the other hand, are broken down into amino acids, some of which can be converted into intermediates that enter the energy pathways. However, this process is less direct and is used primarily when carbohydrate stores are low. What is remarkable about these energy-releasing processes is not just their complexity, but their precision. Every reaction along the way is carefully regulated by enzymes. If any one step were to go awry, the entire energy production process could be disrupted. Enzymes ensure that energy is released in manageable quantities, without excess heat or waste. They control the speed of the reactions, ensuring that energy is made available at the right times, in the right amounts. The beauty of this system is that it is adaptive. When the body requires more energy, such as during exercise or heightened metabolic activity, enzyme activity accelerates, allowing the production of ATP to keep pace with demand. When energy supply outstrips demand, enzymes slow down the production processes, storing excess energy in the form of glycogen or fat for future use. During rest and sleep, energy production slows down as the body transitions into a maintenance mode. Cellular processes that require energy, such as protein synthesis and DNA repair, continue, but at a reduced pace. Enzymes adjust their activity to reflect this change in priority. Instead of fueling muscle contraction or cognitive activity, energy is redirected toward healing and renewal. This quiet work occurs continuously, ensuring that the body is ready for the activity that will begin once wakefulness returns. This ability to store and release energy in response to demand is crucial for survival. The human body does not constantly need the same amount of energy, yet it must always be prepared to respond to varying conditions. Whether it's the sudden need for a burst of energy or the quiet maintenance of bodily functions during sleep, enzymes facilitate this dynamic process, ensuring that energy is always available when needed. In addition to guiding energy production, enzymes also ensure that the body's energy systems are well regulated. Without enzymes, energy production could become erratic, leading to dangerous fluctuations in cellular function. Enzymes prevent this by maintaining balance, adjusting their activity in response to changing conditions, and keeping energy production steady, efficient, and controlled. As you sleep, your body's enzymes continue their work, quietly converting stored nutrients into energy for essential processes. Energy flows through cells, sustaining the maintenance and repair required to keep your body healthy. There is no dramatic surge of energy during sleep, just a quiet, steady production that keeps everything running smoothly. Your body does not stop working while you rest. It simply shifts its focus from outward activity to internal renewal. In the next part, we will explore how this energy is utilized within cells and how enzymes help to harness it for the specific needs of tissues and organs. But for now, it is enough to appreciate that even in sleep, energy production continues as a steady, necessary process, quietly supported by the enzymes that guide the transformation of food into the currency of life. 10. The mitochondrial environment. Within nearly every cell of the body exists a small, carefully regulated space devoted to energy transformation. These spaces are not separate rooms in the familiar sense, but enclosed environments shaped by membranes and chemistry. They are the mitochondria, and within them enzymes carry out some of the most essential and delicately balanced work required for life to continue. Mitochondria are often described as power centers, but this description can suggest something loud or forceful. In reality, the mitochondrial environment is calm, orderly, and precise. Energy is not released in bursts, but in controlled steps, guided by enzymes that ensure efficiency without disruption. This controlled release is what allows cells to meet their energy needs while maintaining stability. The interior of a mitochondrion is distinct from the rest of the cell. It contains its own carefully maintained conditions, including specific concentrations of molecules and a unique arrangement of membranes. These conditions are essential because the enzymes that operate here are finely tuned to this environment. Outside it, their activity would be less effective or even impossible. Energy-rich molecules arrive at the mitochondria already prepared by earlier enzymatic steps. Sugars, fats, and other nutrients have been converted into intermediate forms that can enter mitochondrial pathways smoothly. Once inside, enzymes guide these molecules through a sequence of reactions designed to extract energy gradually and safely. One of the defining features of the mitochondrial environment is its layered structure. An outer membrane provides separation from the rest of the cell, while an inner membrane folds inward, creating a complex surface. Along this surface, enzymes are positioned in specific arrangements, allowing reactions to occur in close proximity. This organization minimizes unnecessary movement and keeps reactive intermediates under control. Within this space, enzymes do not act alone. They function as part of coordinated systems, passing electrons and molecular fragments from one step to the next. Each enzyme performs a small transformation, and together these transformations add up to efficient energy release. No single reaction dominates. Instead, energy emerges from the cumulative effect of many gentle steps. The mitochondrial environment also manages gradients, subtle differences in concentration and charge that store potential energy. Enzymes are essential in creating and maintaining these gradients. Rather than releasing energy directly as heat, they channel it into structured differences that can later be tapped to produce usable energy molecules. This approach reflects a broader theme in biology. Energy is most useful when it is controlled. Sudden releases are wasteful and potentially damaging. Gradual, guided release allows energy to be captured and used productively. Enzymes within mitochondria embody this principle, shaping energy flow to match the cell's needs. Another remarkable feature of the mitochondrial environment is its responsiveness. When energy demand increases, enzymes adjust their activity, allowing pathways to operate more rapidly. When demand decreases, activity slows. This responsiveness occurs without conscious direction, arising naturally from the interaction between enzymes and their surroundings. During periods of rest and sleep, the mitochondrial environment shifts subtly. Energy demand decreases overall, but it does not disappear. Cells still require energy for maintenance, repair, and internal regulation. The calm continuity of mitochondrial function during sleep is essential for recovery. Processes such as protein repair, membrane renewal, and waste clearance all require energy. By providing this energy steadily, mitochondria allow the body to heal and prepare for the next waking period without strain. Mitochondria also possess a degree of independence that reflects their deep evolutionary history. They contain their own genetic material and produce some of their own components. Enzymes are involved in maintaining this internal system, ensuring that mitochondrial function remains reliable across the lifespan of the cell. This semi-autonomous nature adds resilience. Even as cells age or face stress, mitochondria can adapt their enzyme activity to changing conditions. This adaptability helps preserve cellular function over time, contributing to the body's overall endurance. The mitochondrial environment also plays a role in signaling. Changes in energy status can influence enzyme activity in other parts of the cell, coordinating broader responses. In this way, mitochondria do more than produce energy. They help integrate energy availability with cellular behavior. As you rest, countless mitochondria within your cells continue their quiet work. There is no sensation associated with this activity, no awareness of the steady flow of energy being generated. Yet without it, even rest would not be possible. The heart would falter, breathing would slow too far, and the careful balance of the internal environment would be lost. Instead, energy production continues calmly, shaped by enzymes that have evolved to function reliably under gentle conditions. This reliability allows the body to trust its internal processes, to release consciousness into sleep without fear of collapse. In the next part, attention will broaden from the mitochondrial environment to enzymes operating throughout the cell, exploring how energy and materials are managed beyond these specialized spaces. For now, it is enough to recognize mitochondria as quiet chambers of continuity, where enzymes guide energy release with precision and calm, sustaining life through every moment of rest and renewal. 11 enzymes within every cell. Every cell in the body is a small, self-contained world, carrying out countless tasks that together sustain life. Though cells vary widely in shape and function, they all share a common dependence on enzymes. Within each cell, enzymes form a quiet workforce, guiding reactions that maintain structure, manage resources, and allow the cell to respond to its environment with calm precision. A cell may appear simple when viewed from afar, but inside it is densely populated with molecules in constant motion. Proteins are being assembled and disassembled. Membranes are renewed. Signals are received, interpreted, and passed along. None of this activity would be possible without enzymes shaping the flow of chemistry at every step. Enzymes operate throughout the cell, not confined to a single location. Some reside in the fluid interior, known as the cytoplasm, where they guide a wide range of reactions involved in metabolism and maintenance. Others are associated with membranes, embedded in surfaces where they control the movement of molecules into and out of compartments. Still others work within specialized structures, each adapted to the conditions of its immediate surroundings. This distribution allows enzymes to act locally, responding to the needs of specific regions within the cell. Rather than relying on distant control, the cell benefits from immediate feedback. When a particular molecule accumulates in one area, enzymes nearby respond. When levels fall, activity diminishes. This local responsiveness contributes to the cell's overall stability. One of the most important roles enzymes play within cells is in the management of materials. Cells are constantly synthesizing new components while recycling old ones. Proteins that have fulfilled their purpose are broken down into amino acids, which can be reused. Enzymes guide both the breakdown and rebuilding processes, ensuring that resources are conserved and waste is minimized. This recycling is not hurried. It unfolds steadily, allowing the cell to adapt gradually rather than abruptly. Enzymes ensure that breakdown products are handled safely, preventing the accumulation of potentially harmful fragments. At the same time, they guide assembly with accuracy, reducing errors that could compromise function. Enzymes are also central to the cell's ability to maintain its internal environment. Concentrations of ions, small molecules, and energy carriers must remain within narrow ranges. Enzymatic reactions help regulate these levels, adjusting production and consumption as needed. This regulation allows the cell to remain functional even as external conditions change. Communication within the cell also depends on enzymes. Chemical signals often take the form of molecules that must be activated or deactivated to convey information. Enzymes perform these modifications, turning signals on or off at the appropriate times. This controlled signaling allows the cell to coordinate its activities without confusion or delay. The nucleus, which houses genetic material, is another site of intense enzymatic activity. Enzymes here are responsible for copying DNA, repairing damage, and guiding the production of RNA. These processes must be carried out with exceptional accuracy, as errors can have lasting consequences. Enzymes provide the precision required, guiding each step carefully and consistently. Even the structure of the cell depends on enzymatic guidance. Components of the internal framework are assembled and adjusted through enzyme-mediated reactions. These structures give the cell its shape and help organize its contents, contributing to efficient function. As conditions change, enzymes help remodel these structures, allowing flexibility without loss of integrity. Throughout all of this activity, enzymes work silently. There is no sensation associated with their function, no signal sent to awareness, yet their presence is continuous and essential. A cell deprived of functional enzymes would quickly lose its organization, its chemistry becoming disordered and unsustainable. During sleep, cellular enzyme activity does not cease. Instead, priorities shift. Enzymes involved in repair and maintenance become more active, addressing wear accumulated during waking hours. Damaged proteins are replaced, membranes are reinforced, and energy reserves are adjusted. This nighttime activity supports recovery and prepares cells for the demands of the next day. The quiet persistence of enzymes during sleep highlights their role as custodians of continuity. They do not depend on external cues or conscious direction. Their activity is woven into the fabric of cellular life, proceeding as long as conditions allow. This reliability is one of the reasons living systems can afford periods of rest and unconsciousness. Enzymes within cells also contribute to the body's adaptability over time. As cells encounter new conditions, enzyme expression and activity can change gradually. Some enzymes become more prominent, others less so. These shifts allow cells to adjust their behavior while maintaining overall stability. This adaptability is especially important over the lifespan. As the body grows and ages, cellular demands change. Enzymes respond by altering the balance of reactions, supporting development, maintenance, and eventually adaptation to slower rhythms. Through these adjustments, cells remain functional across decades of change. It is remarkable to consider that all of this complexity arises from interactions at the molecular level. Enzymes do not possess awareness or intent, yet together they create systems capable of remarkable coordination. This coordination is not imposed from above, but emerges from countless local interactions guided by structure and chemistry. As you rest, millions of cells throughout your body are engaged in this quiet work. Enzymes guide reactions in muscle cells, nerve cells, skin cells, and countless others, each contributing to the whole. There is no need to monitor this activity or direct it. The system sustains itself, moment by moment. In the next part, attention will turn toward how enzymes help manage potentially reactive molecules, especially those involving oxygen, protecting cells from damage while allowing vital processes to continue. For now, it is enough to recognize that within every cell, a calm and constant enzymatic presence is maintaining order, preserving balance, and quietly keeping you alive as sleep deepens and the body rests. 12 enzymes and oxygen management. Oxygen is both essential and potentially disruptive. It allows cells to extract large amounts of energy from nutrients, supporting complex life, yet its reactive nature means it must be handled with care. Within the body, enzymes take on the quiet responsibility of managing oxygen, ensuring that its benefits are realized while its risks are kept gently in check. It is steady, precise, and continuous. Every breath brings oxygen into the body, where it enters the bloodstream and is carried to tissues. From there, oxygen diffuses into cells, ultimately reaching the mitochondria, where it plays a central role in energy production. Here, oxygen accepts electrons at the end of a long chain of reactions, allowing energy to be released in a controlled manner. Without oxygen, this process would stall, and energy production would become far less efficient. Yet oxygen's ability to accept electrons also makes it reactive. When oxygen interacts prematurely or incompletely with other molecules, it can form reactive byproducts. These molecules, often referred to as reactive oxygen species, are not inherently malicious, but they are highly energetic and can damage proteins, lipids, and genetic material if allowed to accumulate unchecked. Enzymes provide the primary means of control. Some enzymes ensure that oxygen participates only in appropriate reactions, guiding it through well-defined pathways where its reactivity is harnessed rather than unleashed. Others are dedicated to neutralizing reactive byproducts, converting them into stable, harmless forms before they can cause disruption. This enzymatic management allows the body to benefit from oxygen's power without suffering its consequences. The process is not absolute. Small amounts of reactive oxygen species are always present, and they even serve useful roles in signaling and immune defense. What matters is balance, and enzymes are the instruments of that balance. Within cells, enzymes work continuously to monitor and adjust oxygen-related chemistry. When energy production increases, oxygen use rises, and so does the potential for reactive byproducts. Enzymes respond by increasing protective activity, ensuring that stability is maintained even during periods of heightened demand. During rest and sleep, oxygen consumption decreases slightly, reflecting lower overall energy needs. Enzymes adjust accordingly, reducing activity in some pathways while maintaining essential protective functions. Even in this quieter state, oxygen management remains important. Cells continue to respire, and the potential for imbalance never fully disappears. The blood itself is also part of this management system. Enzymes within red blood cells help protect oxygen-carrying molecules from damage, preserving their ability to deliver oxygen efficiently. These enzymatic defenses ensure that oxygen transport remains reliable over the lifespan of the cell. Beyond individual cells, enzymes contribute to oxygen management at the tissue level. In areas where oxygen availability fluctuates, such as muscles during activity and rest, enzymes help cells adapt. When oxygen is plentiful, energy production proceeds smoothly. When it becomes limited, enzymes shift pathways to maintain function while minimizing stress. This adaptability is especially important during transitions, such as falling asleep or waking. As breathing patterns change and metabolic demands shift, oxygen delivery and use must be recalibrated. Enzymes facilitate these adjustments seamlessly, preventing abrupt changes that could disturb cellular stability. The calm effectiveness of oxygen-managing enzymes is often invisible because success feels like normality. When oxygen chemistry is balanced, there is no sensation to report. Breathing feels effortless, energy levels are appropriate, and tissues function smoothly. It is only when balance is lost that awareness arises. Over long periods, the cumulative effect of oxygen-related reactions contributes to aging and wear. Enzymes that manage oxygen help slow this process by reducing unnecessary damage and supporting repair mechanisms. While they cannot prevent change entirely, they shape its pace, allowing life to persist with continuity rather than abrupt decline. This long-term role highlights another aspect of enzymatic management: patience. Enzymes do not aim for perfection, but for sufficiency. They reduce harm to manageable levels, allowing repair systems to keep pace. As you sleep, oxygen continues to flow through your body. Cells continue to use it. Enzymes continue to guide its participation in chemistry, and protective systems remain quietly active. There is no need for awareness or intervention. The process unfolds as it has countless times before. The lungs draw in air. The heart circulates blood, and enzymes at the cellular level ensure that oxygen's journey ends in usefulness rather than disruption. This layered coordination allows sleep to be restorative rather than risky, providing confidence that rest will not compromise vital processes. In the next part, attention will turn toward enzymes involved in repair and renewal, exploring how the body uses periods of rest to restore and maintain itself at the molecular level. For now, it is enough to appreciate oxygen not as a threat to be feared, but as a powerful resource carefully managed by enzymes that work without pause, preserving balance and calm through every breath, every moment, and every quiet hour of sleep. 13. Repair, renewal, and maintenance. Life persists not because structures never wear down, but because renewal quietly keeps pace with change. Within the body, nothing is truly static. Proteins age, membranes experience stress, and genetic material encounters small imperfections, simply through the act of existing. What allows life to continue smoothly is not the absence of damage, but the presence of constant repair. Enzymes are at the center of this restorative process, guiding maintenance with patience and precision. Every cell carries within it the tools needed to monitor its own condition. Enzymes participate in systems that detect irregularities, identify worn components, and initiate gentle correction. This process is continuous, unfolding without awareness, ensuring that small problems are addressed before they accumulate into larger disruptions. Proteins, for example, are essential working molecules, but they are not permanent. Over time, their shapes can be altered by use, temperature shifts, or chemical interactions. Enzymes help recognize proteins that are no longer functioning optimally. Once identified, these proteins are broken down into their basic components, which can then be reused to build new, properly functioning proteins. This recycling conserves resources and maintains cellular efficiency. The renewal of proteins is not rushed. Enzymes guide breakdown and rebuilding at rates that preserve balance. Too rapid a turnover would waste energy, while too slow a process would allow malfunction to spread. Enzymes ensure that renewal proceeds at a pace that matches need. Membranes also require constant attention. These thin boundaries define cells and compartments, controlling what enters and exits. Their components are exposed to physical and chemical stress, especially in active tissues. Enzymes support the renewal of membrane molecules, replacing damaged parts and maintaining flexibility. This maintenance preserves the cell's ability to interact with its environment smoothly. Even genetic material, often thought of as stable, benefits from enzymatic care. Enzymes dedicated to repair patrol genetic material, correcting errors and restoring integrity. These corrections are usually small and precise, preventing the gradual accumulation of changes that could interfere with cellular function. The effectiveness of these repair systems is especially evident during sleep. When external demands decrease, the body redirects energy and attention inward. Enzymes involved in maintenance become more active, addressing wear accumulated during waking hours. This shift allows restoration to proceed without competing demands, making sleep a vital context for renewal. Throughout the night, enzymes guide the replacement of worn molecules, the reinforcement of structures, and the balancing of chemical conditions. There is no sensation associated with this work, no signal to consciousness. Yet by morning, the cumulative effect of these quiet actions supports a feeling of readiness and stability. Repair is not limited to individual molecules. Entire cellular systems undergo maintenance. Enzymes help regulate processes that remove damaged cells and support the creation of new ones. This turnover allows tissues to remain functional over long periods, even as individual cells come and go. This approach to maintenance reflects a broader biological philosophy. Rather than striving for permanence, life embraces change, provided it is guided and controlled. Enzymes embody this philosophy by facilitating continuous renewal. They do not attempt to freeze structures in place, but to preserve function through gradual replacement. Over the course of a lifetime, this balance between wear and repair shapes aging. While enzymes cannot prevent change entirely, they influence its pace and character. By supporting effective maintenance, they allow tissues to remain resilient for decades. Aging becomes a process of adjustment rather than sudden decline. The calm nature of enzymatic repair also contributes to resilience under stress. When conditions challenge the body, repair systems can adapt, increasing activity where needed. Once conditions return to normal, activity subsides. This flexibility allows the body to recover from fluctuations without losing stability. Enzymes involved in repair also coordinate with other systems. Energy availability, oxygen management, and nutrient supply all influence maintenance activity. Enzymes integrate these signals, ensuring that repair proceeds when resources are sufficient and slows when conservation is needed. This coordination prevents strain and preserves balance. As you rest, these systems are active throughout your body. Muscles, skin, organs, and even the brain benefit from ongoing maintenance. Enzymes guide each step quietly, without interruption, supporting the integrity of tissues that must function again upon waking. The idea that the body repairs itself continuously can be reassuring. It suggests that life is supported not by fragile perfection, but by robust processes designed to accommodate imperfection. Enzymes make this accommodation possible, smoothing the effects of daily wear and allowing recovery to become routine. This quiet renewal is not something that needs encouragement or awareness. It has been unfolding since before birth, guided by molecular systems, refined over evolutionary time. Sleep simply provides a context in which these systems can work with fewer distractions. In the next part, attention will turn toward how enzymes participate in the nervous system, supporting communication and balance within the brain and nerves. For now, it is enough to rest in the knowledge that while the mind drifts, enzymes are at work throughout the body, repairing, renewing, and maintaining the structures that make life possible, carrying continuity gently forward through every quiet hour of sleep. 14 enzymes and the nervous system. The nervous system is often described in terms of speed. Signals travel rapidly, responses feel immediate, and thoughts seem to arise in an instant. Yet beneath this apparent quickness lies a foundation of calm, deliberate chemistry. Every signal passed along a nerve, every sensation registered, and every movement initiated depends on enzymes working quietly behind the scenes, shaping and regulating the chemical environment that makes neural communication possible. Neurons, the primary cells of the nervous system, are specialized for communication. They transmit information using electrical changes along their membranes and chemical signals at their connections. While the electrical aspect often draws attention, it is the chemical side that relies most heavily on enzymes. Without enzymatic guidance, neural signals would blur, persist too long, or fail to end when they should. At the junctions between neurons, known as synapses, communication occurs through chemical messengers called neurotransmitters. These molecules are released from one neuron, cross a small gap, and bind to receptors on the next cell. This process allows information to move from cell to cell, forming the basis of perception, thought, and action. Enzymes are essential at every stage of this exchange. They help synthesize neurotransmitters from simpler precursors, ensuring that adequate supplies are available. They assist in packaging these molecules into tiny vesicles, preparing them for release. And once a signal has been transmitted, enzymes help clear neurotransmitters away, ending the message cleanly and restoring readiness for the next signal. This clearing process is especially important. If neurotransmitters linger too long in the synapse, signals can become prolonged or distorted. Enzymes prevent this by breaking down neurotransmitters or converting them into inactive forms. This allows neural communication to remain precise, with clear beginnings and endings. The balance maintained by these enzymes contributes to mental clarity and emotional stability. Instead, they flow in patterns shaped by both experience and internal chemistry. Enzymes ensure that these patterns remain flexible without becoming chaotic. Within neurons themselves, enzymes manage energy use and maintenance. Nerve cells are metabolically active, requiring continuous energy to maintain electrical gradients and support signaling. Enzymes guide the pathways that supply this energy, adjusting activity according to demand. During periods of intense thought or movement, energy production increases. During rest, it subsides gently. The nervous system also depends on enzymes for protection. Neural tissue is sensitive and must be shielded from chemical imbalance. Enzymes help regulate ions and remove potentially harmful byproducts, preserving the delicate environment required for signaling. Sleep highlights the importance of enzymatic balance in the nervous system. As consciousness fades, patterns of neural activity change. Some circuits become less active, while others involved in memory consolidation and maintenance remain engaged. Enzymes adjust to these shifts, supporting the altered chemistry of the sleeping brain. During sleep, enzymes help regulate neurotransmitter levels associated with wakefulness and rest. Chemical messengers that promote alertness diminish, while those associated with calm and restoration become more prominent. This transition is gradual and smooth, guided by enzymatic activity rather than abrupt switches. Enzymes also participate in processes that reinforce neural connections. Experiences from the day leave subtle chemical marks within the brain. During sleep, contributing, this reinforcement occurs quietly, without awareness, shaping future responses and understanding. The calm precision of enzymatic action in the nervous system is especially evident in how quickly balance can be restored after disturbance. Emotional stress, intense thought, or sensory overload can alter neural chemistry. Enzymes work to bring conditions back toward equilibrium, clearing excess signals and replenishing depleted resources. Over the lifespan, enzymes help the nervous system adapt to change. Development, learning, and aging all involve shifts in neural chemistry. Enzymes respond by adjusting synthesis, breakdown, and recycling processes, supporting flexibility while preserving core function. This adaptability underscores a deeper truth about the nervous system. While it feels immediate and responsive, it is grounded in slow, careful chemical management. Enzymes provide the patience that underlies speed, ensuring that rapid signals do not lead to instability. As you rest, your nervous system remains active in a different mode. Sensory input decreases, voluntary movement pauses, and attention withdraws from the external world. Yet enzymes continue to manage neural chemistry, maintaining balance, supporting repair, and preparing circuits for future activity. There is comfort in knowing that thought and awareness can safely recede because the underlying system remains stable. Enzymes do not require vigilance. They continue their work automatically, preserving the conditions that make waking consciousness possible. In the next part, attention will turn toward how enzymes interact with hormonal signaling and long-term regulation, exploring another layer of quiet coordination within the body. For now, it is enough to recognize that the nervous system, for all its apparent speed and complexity, rests upon a foundation of calm enzymatic guidance, working continuously to maintain clarity, balance, and the gentle rhythms that carry you through sleep and waking alike. 15 hormones, signals, and timing. Beyond the rapid exchanges of the nervous system lies another layer of communication. This is the hormonal system, a network of chemical signals that travel through the bloodstream, influencing cells far from their point of origin. While hormones themselves often take center stage in descriptions of this system, enzymes are the quiet organizers that give hormonal signaling its timing, precision, and restraint. Hormones are released by specialized cells and glands, entering circulation and drifting throughout the body. Unlike nerve signals, which act almost instantly and locally, hormonal signals unfold over minutes, hours, or even days. This slower pace allows them to coordinate long-term processes such as growth, metabolism, sleep-wake rhythms, and adaptation to changing conditions. Enzymes ensure that this slowness remains purposeful rather than vague. The life of a hormone begins with synthesis. Most hormones are not created spontaneously but are assembled through stepwise reactions guided by enzymes. Raw molecular materials are gradually transformed into specific signaling molecules, each step controlled to prevent excess or deficiency. Enzymes determine how quickly hormones are produced and in what quantities, shaping the strength and duration of the signal even before it is released. Once a hormone enters the bloodstream, enzymes continue to influence its journey. Some enzymes modify hormones into active forms, while others convert them into inactive variants. This conversion allows the body to fine-tune signaling without altering release itself. A hormone may circulate widely, but only become active where specific enzymes are present, adding another layer of spatial control. Timing is central to hormonal function. Many hormonal signals follow daily or seasonal rhythms, rising and falling in predictable patterns. Enzymes participate in maintaining these rhythms by regulating synthesis and breakdown. When the time for a signal has passed, enzymes help clear hormones from circulation, preventing them from lingering longer than intended. This clearing process is as important as release. Prolonged hormonal signals can disrupt balance, leading to overstimulation or suppression of target tissues. Enzymes prevent such disruptions by breaking hormones down into harmless components once their message has been delivered. This ensures that signaling remains cyclical rather than cumulative. Within target cells, enzymes play yet another role. Hormones often act by triggering internal chemical cascades, amplifying their message inside the cell. Enzymes guide these cascades, turning small external signals into coordinated internal responses. Just as importantly, other enzymes bring these responses to an end, restoring the cell to a resting state. The coordination between hormones and enzymes allows the body to integrate information over time. Signals related to energy availability, stress, growth, and repair are blended together, shaping cellular behavior in ways that reflect both immediate conditions and long-term needs. Enzymes act as interpreters, translating chemical messages into appropriate actions. Sleep provides a clear example of this coordination. Signals associated with alertness diminish, while those supporting repair and conservation rise. Enzymes guide these transitions, adjusting hormone levels gradually so that sleep arrives smoothly rather than abruptly. Throughout the night, enzymes continue to regulate hormonal balance. Some hormones reach their peak activity during sleep, supporting tissue repair, immune function, and metabolic adjustment. Others are reduced, allowing the body to disengage from external demands. These shifts occur reliably night after night, guided by enzymatic timing mechanisms that require no conscious oversight. The interaction between enzymes and hormones also allows flexibility. When routines change, such as during travel or altered sleep schedules, enzymatic regulation helps the body adjust. Hormonal rhythms may shift temporarily, but enzymes work to reestablish balance, smoothing the transition and minimizing disruption. Over longer timescales, enzymes shape hormonal changes across the lifespan. Puberty, maturity, and aging all involve shifts in hormonal patterns. Enzymes adjust synthesis and breakdown rates gradually, allowing the body to adapt without sudden imbalance. These changes are part of a continuous process rather than a series of abrupt stages. Stress offers another illustration of enzymatic timing. Hormones released in response to challenge prepare the body for action, but they must also be cleared once the challenge passes. Enzymes ensure that stress signals do not persist unnecessarily, allowing the body to return to a calm baseline. This ability to resolve signals is as important as the ability to initiate them. The calm effectiveness of enzymatic regulation in the hormonal system often goes unnoticed because it prevents extremes. When timing is right, the body feels steady. Energy levels fluctuate within comfortable ranges. Sleep comes when expected, and recovery follows exertion naturally. Enzymes are the reason these patterns hold. As you rest, hormonal signaling continues quietly. Enzymes adjust levels, convert signals, and clear messages that have served their purpose. There is no urgency in this work, only continuity. The body uses the stillness of sleep to reinforce rhythms that will guide the coming day. This layered communication system reflects a broader truth about life. Not all guidance needs to be fast. Some processes benefit from slowness, from signals that unfold over time and influence many systems at once. Enzymes allow this slowness to remain orderly, preventing drift and preserving coherence. In the next part, attention will turn toward regulation itself, exploring how enzyme activity is adjusted automatically through feedback mechanisms that require no awareness or instruction. For now, it is enough to recognize hormones and enzymes as partners in timing, shaping the long, quiet arcs of physiological change that continue even as consciousness fades, carrying the body gently through rest and renewal. 16. Regulation without awareness. One of the most remarkable features of the living body is that it does not require supervision to remain balanced. While the mind drifts through thought, attention, and sleep, the body continues to regulate itself with quiet consistency. At the center of this self-regulation are enzymes, adjusting chemical activity moment by moment, without instruction, intention, or awareness. Life persists not because it is constantly watched, but because regulation is built into its chemistry. Regulation begins with a simple principle. Reactions respond to conditions. Enzymes are sensitive to their immediate environment, and this sensitivity allows them to act as natural regulators. When a molecule becomes abundant, enzymes that interact with it encounter it more often, increasing reaction rates. When the molecule becomes scarce, reactions slow. This automatic responsiveness forms the basis of regulation throughout the body. This process requires no central command. Instead, regulation emerges from countless local interactions, each enzyme responding only to what is present nearby. The combined effect of these small adjustments is a stable whole, maintained without effort. Feedback plays a central role in this system. Many enzymatic pathways include built-in feedback mechanisms, where the end product of a sequence influences earlier steps. When enough product has been made, it interacts with enzymes upstream, gently reducing their activity. When product levels fall, this inhibition lifts and production resumes. This feedback keeps reactions within appropriate bounds. Such feedback loops are not rigid controls. They are flexible, allowing slight fluctuations while preventing extremes. This flexibility is essential for adapting to changing conditions. The body does not aim for perfect constancy, but for values that remain within safe and functional ranges. Regulation without awareness is especially evident in energy management. During sleep, when demand is lower, energy pathways shift toward maintenance and conservation. The same principle applies to temperature regulation, nutrient balance, and chemical composition. Enzymes involved in these processes adjust continuously, responding to shifts in concentration, pH, and energy availability. This constant adjustment creates a sense of stability that feels effortless from the inside. What makes this system especially robust is redundancy. Often, multiple enzymes or pathways can contribute to the same outcome. If one route slows, another may compensate. This redundancy reduces vulnerability, allowing regulation to continue even when individual components are stressed or impaired. Sleep provides a clear demonstration of regulation without awareness. As consciousness fades, the body does not pause to await instruction. Breathing continues, heart rhythm remains steady, and internal chemistry adjusts to a restorative mode. Enzymes guide these transitions smoothly, ensuring that rest does not compromise essential function. During sleep, regulatory systems focus on restoration rather than responsiveness to the external world. Enzymes involved in repair, waste clearance, and resource redistribution become more prominent. Feedback mechanisms ensure that these processes proceed efficiently without overuse of energy. This unconscious regulation is not limited to sleep. It operates continuously, supporting every moment of wakefulness as well. Whether you are active or still, calm or stressed, enzymes adjust chemistry to support the current state while preserving long-term balance. The nervous and hormonal systems both rely on enzymatic regulation to function properly. Signals must be initiated, amplified, and terminated at the right times. Enzymes ensure that signals do not linger unnecessarily or escalate beyond control. This regulation supports clarity, emotional balance, and physiological stability. Over longer periods, regulation without awareness shapes adaptation. New patterns emerge without conscious planning, allowing the body to accommodate novelty while maintaining continuity. This capacity for self-regulation reflects evolutionary refinement. Systems that required constant oversight would be fragile and inefficient. By embedding regulation into chemistry itself, life has achieved resilience. It can be comforting to recognize how little intervention is required to stay alive. Even complex coordination arises naturally from simple principles repeated at scale. Enzymes do not think, decide, or intend. Yet together they create systems capable of remarkable stability and adaptability. As you rest, regulation continues quietly. Blood chemistry remains within narrow limits. Energy is allocated appropriately. Repair processes unfold at measured rates. None of this requires awareness. The body carries itself forward through the night with confidence built into its molecular design. In the next part, attention will turn toward the role of nutrients and cofactors, exploring how enzymes rely on subtle partnerships to function optimally. For now, it is enough to rest in the understanding that life is not held together by constant effort, but by regulation woven into chemistry itself, working steadily and silently, keeping you alive while you sleep. 17. Nutrition and enzyme support. Enzymes, for all their precision and reliability, do not work entirely alone. Many depend on quiet partnerships with small molecules that help them maintain shape, guide reactions, or carry chemical groups from one place to another. These partnerships are subtle, often overlooked, yet essential. Nutrition provides many of these supporting elements, linking the outside world gently to the inner chemistry that sustains life. Food is often thought of primarily as a source of energy or building material, but at a deeper level, it also supplies the fine-tuning components that allow enzymes to function optimally. These components do not fuel reactions directly. Instead, they assist enzymes in doing their work smoothly and accurately, preserving balance rather than driving force. Some enzymes require the presence of small helper molecules, often referred to as cofactors. These may be metal ions or organic molecules that bind temporarily to the enzyme, completing its functional form. Without them, the enzyme may remain inactive or only partially effective. With them, the enzyme's shape and chemical properties align precisely with its task. Many of these cofactors come from the diet. Minerals such as iron, magnesium, zinc, and copper participate quietly in countless enzymatic reactions. They help stabilize structures, facilitate electron movement, or activate specific sites within enzymes. Though needed only in small amounts, their absence can subtly alter enzyme activity across multiple systems. Organic cofactors often originate from vitamins. These molecules are not used as fuel or structural components, but as facilitators of chemical change. Once inside the body, they are often converted into active forms that associate with enzymes, enabling reactions that would otherwise proceed too slowly or not at all. What is striking about these relationships is their economy. A single vitamin-derived cofactor can assist an enzyme through many cycles of activity, much like the enzyme itself. This reuse allows small amounts of nutritional input to support large volumes of biochemical work, reinforcing the efficiency of living systems. Nutrition, in this context, is not about excess. It is about sufficiency and balance. Enzymes do not require constant abundance of cofactors, but they do rely on steady availability. The body manages this by storing certain nutrients and recycling others, ensuring that enzyme support remains consistent even between meals. During digestion and absorption, enzymes help process nutrients into forms suitable for these supportive roles. Once absorbed, nutrients are distributed to tissues according to need. Enzymes involved in transport and storage ensure that cofactors reach the enzymes that depend on them, maintaining harmony across systems. Sleep again provides an important context. During rest, the body reassesses and redistributes resources. Enzymes involved in maintenance may draw upon stored nutrients, using them to support, repair, and renewal. This nighttime usage highlights why nutrition supports not only activity, but also recovery. The relationship between enzymes and nutrition is dynamic. When dietary patterns change, enzyme activity can adjust gradually. Some enzymes become more prominent, others less so. This adaptability allows the body to function across a range of conditions, reflecting flexibility rather than rigidity. It is also important to recognize that the body prioritizes enzyme support. Enzymes involved in basic maintenance and survival receive support before those associated with less critical functions. This prioritization reflects an underlying hierarchy shaped by evolution. Over time, consistent nutritional support contributes to enzymatic stability. Enzymes maintain their structure more effectively, pathways operate smoothly, and regulatory systems remain responsive. This does not mean that nutrition eliminates variability or change. The calm way in which nutrition influences enzyme function mirrors the overall tone of biochemical life. There are no sudden transformations or dramatic shifts under normal conditions. Instead, small inputs support continuous processes, reinforcing balance rather than forcing outcomes. As you rest, nutrients absorbed earlier continue to play their roles. Cofactors associate with enzymes, reactions proceed, and maintenance continues. There is no awareness of this support, no signal that nutrition is at work. Yet without it, enzymatic systems would gradually lose their precision. This quiet dependency connects the external world to the inner one in a gentle loop. Food becomes molecules. Molecules become partners in chemistry, and chemistry sustains the body that will one day seek food again. Enzymes sit at the center of this loop, translating material availability into living continuity. In the next part, attention will turn toward how enzyme activity changes gradually across the lifespan, adapting to growth, maturity, and aging. For now, it is enough to recognize nutrition not as a force that drives the body, but as a subtle support that allows enzymes to keep working calmly, reliably, and without interruption, even as sleep deepens and the body rests in quiet balance. 18 enzymes across the lifespan, from the earliest moments of development to the quiet years of later life. Enzymes remain constant companions. They do not mark time or track age, yet their activity subtly shifts as the body grows, adapts, and changes. These shifts are not sudden or dramatic. They unfold gradually, reflecting the body's ability to adjust its chemistry to different stages of life while preserving continuity. In early development, enzymatic activity supports rapid growth and organization. Cells divide frequently, tissues form, and systems establish their basic patterns. Enzymes guide the construction of proteins, membranes, and genetic material with high efficiency. During this time, many pathways operate at heightened activity, supplying the materials and energy needed for expansion and differentiation. As growth slows and maturity is reached, enzymatic patterns stabilize. The focus shifts from construction to maintenance. Enzymes continue to support repair, energy management, and regulation, but at rates suited to a body no longer expanding. This transition is smooth, guided by changes in enzyme expression and activity that occur gradually over time. Throughout adulthood, enzymes help maintain equilibrium. Daily wear is balanced by nightly repair. Energy demands fluctuate with activity and rest, and enzymes adjust accordingly. The overall pattern remains steady, supporting resilience and adaptability. With advancing age, enzymatic activity does not simply decline. Instead, it changes character. Some pathways become less active, others more so. Repair processes may proceed more slowly, while protective mechanisms become increasingly important. These shifts reflect adaptation rather than failure, allowing the body to function effectively under changing conditions. One of the defining features of enzymatic aging is variability. Not all tissues change at the same rate, and not all enzymes are affected equally. Some retain their activity well into later life, continuing to support essential functions. Others adjust their roles, contributing to new patterns of balance. Enzymes involved in energy production, for example, may operate at slightly reduced capacity, reflecting lower overall demand. At the same time, enzymes involved in maintenance and protection may become more prominent, helping preserve stability. This redistribution supports longevity by prioritizing essential functions. The nervous system also reflects these gradual changes. Enzymes that manage neurotransmitters and neural maintenance adjust activity subtly, supporting continued communication while accommodating slower rhythms. Cognitive function remains possible because enzymatic balance is preserved, even as patterns shift. Sleep continues to play a vital role across the lifespan. Enzymes continue to guide renewal, even if the pace differs from earlier stages. This continuity supports the sense of self that persists through change. It is important to recognize that aging is not driven by a single process. It emerges from the cumulative effects of countless small changes. Enzymes influence these changes by shaping how chemistry responds to time, stress, and environment. They cannot prevent all change, but they modulate its expression. Nutrition, environment, and activity all interact with enzymatic aging. This responsiveness allows individuals to maintain function under diverse conditions, reflecting flexibility rather than rigidity. The calm nature of enzymatic adaptation is often overlooked because it lacks drama. Instead, there is continuity, shaped by gradual shifts in chemical emphasis. Enzymes provide the means for this continuity, allowing life to remain coherent as circumstances evolve. From a broader perspective, enzymes represent the body's commitment to persistence. They do not strive for youth or resist change. They support function in the present moment, whatever that moment requires. This focus on immediacy rather than ideal states allows life to continue with dignity and balance. As you rest, your enzymes reflect your current stage of life. They are not comparing it to the past or anticipating the future. They are responding to now, guiding chemistry in ways that support stability and continuity. This present-focused activity is one of the reasons rest feels restorative at any age. In the final parts of this series, attention will turn toward the long evolutionary history that shaped these enzymes and the quiet closing reflections that bring the journey full circle. For now, it is enough to recognize enzymes as lifelong companions, adapting gently as time passes, preserving balance, and sustaining life through every stage, every night, and every quiet breath of sleep. 19 Evolution's slow refinement. Long before enzymes worked quietly within human cells, they were already shaping the chemistry of life in far simpler forms. Their story stretches back through deep time, across countless generations of organisms adapting to their environments. Enzymes did not appear all at once, fully formed and precise. They emerged gradually, refined through slow experimentation, preserved not by intention, but by usefulness. In the earliest stages of life, chemical reactions occurred with little guidance. Simple molecules interacted in basic ways, driven by the conditions of their surroundings. Over time, Certain molecular structures began to influence these reactions, making some pathways more likely than others. Molecules that could gently guide chemistry gained an advantage because they allowed reactions to proceed more reliably under mild conditions. These early catalytic molecules were not yet enzymes as we know them today. They were simpler, less specific, and less efficient, but even modest guidance was enough to confer stability. Systems that could maintain their chemistry were more likely to persist, replicate, and diversify. Over vast spans of time, this persistence shaped the emergence of increasingly refined enzymes. As life grew more complex, so too did its chemistry. Cells developed internal compartments, energy demands increased, and reactions became more interconnected. This growing complexity required greater precision. Enzymes adapted by becoming more selective, more efficient, and more responsive to their environments. Structures that supported accurate folding and reliable function were preserved and passed on. Evolution did not aim for perfection. It favored sufficiency. Enzymes that worked well enough under given conditions were retained, even if they were not optimal in every context. This principle allowed life to remain flexible, adapting to new environments without discarding existing systems entirely. One of the most striking outcomes of this slow refinement is the unity of enzymatic chemistry across living organisms. Many enzymes found in humans are remarkably similar to those in plants, animals, and even single-celled organisms. This similarity reflects shared ancestry and the effectiveness of certain solutions once they arise. For example, the basic pathways that manage energy, repair genetic material, and assemble proteins are conserved across diverse forms of life. Enzymes that perform these tasks have changed little over immense periods of time, because their roles are fundamental and their solutions effective. This conservation speaks to the deep logic underlying biological chemistry. At the same time, variation exists. Enzymes have adapted to specific environments, temperatures, and chemical conditions. Organisms living in extreme environments possess enzymes suited to those conditions, demonstrating flexibility built upon a shared foundation. Evolution balances stability with adaptability, preserving core functions while allowing contextual refinement. The refinement of enzymes also contributed to the emergence of multicellular life. As organisms grew larger and more complex, coordination between cells became essential. Enzymes supported this coordination by enabling controlled signaling, energy distribution, and maintenance across tissues. Without enzymatic precision, such coordination would have been impossible. Over evolutionary time, enzymes also became integrated into regulatory networks. Their activity could be influenced by other molecules, creating feedback systems that enhanced stability. These networks allowed organisms to respond to changes without losing coherence, supporting survival in dynamic environments. Importantly, evolution shaped enzymes without foresight. The remarkable suitability of enzymes to modern life is the cumulative result of countless small steps, each shaped by local conditions and immediate outcomes. This perspective adds a sense of calm inevitability to enzymatic function. Enzymes are not fragile constructs requiring constant correction. They are robust outcomes of long refinement, designed to work reliably across a range of conditions. This robustness is one of the reasons life can sustain itself through rest, stress, and change. As you sleep, the enzymes within you are the inheritors of this long history. Their shapes and behaviors carry echoes of ancient chemistry, refined through unbroken lines of descent. They do not strive or innovate in the moment. They simply perform the roles they have been shaped to perform. The evolutionary history of enzymes also reminds us that life is grounded in continuity. The same chemical principles that operated billions of years ago are at work now, quietly guiding reactions in every cell. This continuity connects the present moment to deep time, linking individual rest to the long persistence of life. Understanding enzymes through an evolutionary lens also reframes vulnerability. When enzymatic systems falter, it is not because they are poorly designed, but because they are operating under conditions beyond those in which they were refined. Even then, redundancy and adaptability often preserve function. Evolution's slow refinement has produced systems that do not require perfection to endure. They require balance, responsiveness, and continuity. Enzymes embody these qualities, allowing life to persist through gradual change rather than abrupt transformation. As this exploration nears its close, it becomes clear that enzymes are not merely tools within the body. They are expressions of life's long negotiation with chemistry, shaped to support stability, adaptability, and quiet persistence. They carry the memory of countless past lives in their structure and function. In the final part, attention will turn inward again, drawing these themes together into a calm reflection on the body as a steady biochemical system. For now, it is enough to rest in the knowledge that the enzymes working within you tonight are the result of deep time, refined patiently, sustaining life with the same quiet effectiveness that has carried it forward for billions of years. 20. A quiet closing. Reflection. As this long, gentle journey draws to a close, it returns naturally to stillness. After exploring enzymes across digestion, energy, repair, signaling, and deep time, what remains is not a list of processes, but a sense of continuity. Life, when viewed through the lens of enzymes, appears less dramatic and more dependable. It is not sustained by bursts of effort, but by steady guidance unfolding moment by moment. Within you, right now, enzymes are working exactly as they have throughout this entire listening experience. They have not paused for attention, nor have they required understanding. Their presence is constant, woven into the background of being alive. While thoughts drift and awareness softens, chemistry continues with quiet assurance. The body can be imagined as a vast, calm landscape rather than a machine. In this landscape, enzymes are not operators pulling levers, but natural features shaping the flow of events. Like riverbanks guiding water, they do not create motion, but give it direction. Without strain or urgency, they ensure that change remains compatible with life. Every breath you take is supported by enzymes managing oxygen. Every heartbeat depends on energy released gently, step by step. Every cell relies on repair systems that quietly replace what is worn down. None of these processes require awareness. They do not ask for approval or instruction. They simply continue, refined through time, reliable through rest. This reliability is what allows sleep to be safe. When consciousness loosens its hold, the body does not lose coherence. Instead, it leans more fully into systems designed to function without observation. Enzymes become even more central during these hours, supporting maintenance, balance, and renewal while the mind withdraws. The stillness of sleep is not emptiness. Enzymes ensure that this activity remains harmonious, preventing excess and correcting small deviations before they become significant. This quiet vigilance allows rest to restore rather than disrupt. Seen from this perspective, life is not fragile in the way it sometimes appears. It is supported by layers of regulation, redundancy, and balance. Enzymes embody these qualities. They do not seek perfection, only sufficiency. They do not resist change, but shape it gently. Across the lifespan, enzymes adapt without judgment. They support growth, maturity, and aging with equal steadiness. They do not cling to earlier patterns or rush toward new ones. They respond to what is present, guiding chemistry in ways that preserve continuity. This present-focused nature of enzymatic activity offers a subtle lesson. The body does not live in memory or anticipation. It lives in the current state of molecules, conditions, and needs. Enzymes respond to now, and in doing so, They carry life forward one moment at a time. The evolutionary history of enzymes reinforces this sense of calm persistence. These molecules are not recent inventions, nor are they easily undone. They are the result of deep refinement, shaped by survival rather than design. Their effectiveness lies in their simplicity and reliability. Even complexity emerges quietly through them. Vast networks of reactions remain stable because each individual step is guided carefully. Together, they create a system where life unfolds smoothly rather than abruptly. As you rest, there is nothing you need to do to support this system. No adjustment is required. No understanding is demanded. The body does not wait for readiness or clarity. It continues, gently and confidently, supported by chemistry that has been unfolding since long before conscious thought existed. This closing moment is not an ending so much as a return to awareness of what has always been present. Enzymes have been at work since before this narration began, and they will continue long after it fades. Their constancy is what allows experience to come and go without threatening the foundation beneath it. In the quiet of sleep, this foundation remains intact. Cells maintain themselves. Balance is preserved. Energy flows at the pace required for rest. The body holds itself together without effort. If there is comfort to be found in biochemistry, it lies here. Life is not maintained by vigilance, but by systems designed to function without it. Enzymes do not hurry, struggle, or exhaust themselves. They repeat their work calmly, reliably, and endlessly, allowing you to let go. As listening softens and awareness drifts, nothing essential is left unattended. The chemistry that keeps you alive continues its quiet work, moment by moment, breath by breath. There is steadiness beneath stillness, motion beneath rest. And so, as this exploration gently settles, it leaves you not with information to remember, but with reassurance. Inside you, countless enzymes continue to guide life forward, calmly and faithfully. Through sleep, through waking, and through the long, uninterrupted flow of being alive.